Hi everyone, welcome to our third week of classes dedicated to methods in cognitive neuroscience. However, before we get started on that topic, I want to finish up with a few notes, a few leftover slides from last week. And the topic that we concluded our discussion with last week was about the role of the skull in protecting the brain. And that cannot be overstated. However, the skull is just one level of protection for the brain from adverse mechanical forces. Another important mechanism that protects the brain is provided by the ventricular system, or to be more precise, by the cerebrospinal fluid contained within the ventricular system. So what does this do and how does it function? Well, the ventricular system is the result of four different interconnecting cavities, the ventricles, placed within the brain. To be more specific, two lateral ventricles, one for each hemisphere, which run all the way from the frontal lobe to the back of the brain to the occipital lobe, along with two smaller ventricles, one central placed between the two thalami, and one placed in between the cerebellum and the brainstem. Okay, so all these ventricles are interconnected and allow the free passage of cerebrospinal fluid that they are filled with. Perhaps more interestingly is the fact that the same cerebrospinal fluid, CSF, is also the one that surrounds the brain. To be more precise, surrounds the pia mare that encloses the brain and freely flows in between the pia mare and the arachnoid matter. Also, the same CSF passes into the spine, so you can finally get some kind of impression of the size and significance of this interconnected systems responsible for producing and for allowing the passage of CSF. Okay, but what is its actual function? Well, as the title implies, it is responsible for protecting the brain from mechanical forces such as rapid acceleration and deceleration, as well as impact to the brain, as you sustain, for instance, during a concussion. So the cerebrospinal fluid and all these channels and ventricles that enclose it are responsible for cushioning the brain from rapid movements. Another way in which the CSF protects the brain is from its own weight. And this might seem curious at first because the brain is not such a large or heavy structure. The average weight of the adult human brain is around 1,400 grams. So while this might not seem like a lot, it is enough to block the passage of blood, or at least reduce it by placing a lot of pressure on some of the cerebral arteries, and therefore blocking the blood supply to some parts of the brain, leading to neuronal death. Well, this would be a very unfortunate outcome. And to counter the weight of the brain, the CSF places it within a state of buoyancy. So to be more to provide a more intuitive term for that, the brain essentially floats within cerebrospinal fluid, which reduces the overall buoyancy and the weight placed on some of the cerebral arteries. So in that way, the ventricular system and the CSF provide protection for the brain, both from outside forces and from the brain itself and its own weight. And this conveniently leads us to the next topic, which is cerebral vasculature. At this point, we should already have an appreciation for how important blood supply is to the brain. Blood is responsible for supporting the high metabolic cost of brain functioning. We do know that the brain is responsible, or rather, it's taking up about 20% of the oxygen that we take in, which is a considerable proportion. And an uninterrupted normal blood supply to the brain is a key to ensuring that that constant necessary supply of oxygen is delivered in the right regions. Okay, so how does this happen? Well, all this blood is being relayed to the brain through a number of arteries, some running along the front of the neck, the carotid arteries, and some running along the spinal cord, the vertebral arteries. And all of them come together at the base of the brain into this circled area right here, the circle of Lewis, from which they are further extended, spread out across a number of cerebral arteries, carrying blood to different networks of regions of the brain. And we're only going to look at three of the major ones, starting with the largest of them. That will be the middle cerebral artery, which projects here from the temporal lobe on the lateral side of the brain to the front, the posterior, and even the occipital lobe, its lateral side. And you can appreciate the extent to which this middle cerebral artery provides blood supply to cortical matter. Okay, a mid-sagittal section through the brain reveals two other major cerebral distributions. One, the anterior cerebral artery running from the frontal lobe through the posterior lobe, and the other one from the mid-temporal lobe through the occipital lobe. Okay, so why are we talking about this? Well, it's not just because blood is important to the brain, but also because of the specific and predictable outcomes of stopping or preventing blood from normal flow within these arteries. And we had a brief discussion about stroke last week. And the outcome of blocking blood flow through a specific artery. Well, you can imagine, for instance, that if the posterior cerebral artery is damaged through blockage or by erupting, by bursting, then a lot of the blood supply that flows to the back of the brain, the occipital lobe, will not be available any longer. Now, since the occipital lobe is responsible for visual processing, then the predictable outcome is visual problems, visual impairments. So this is how the anatomy and this is how the overall architecture of cerebral distributions has very concrete outcomes when we start to discuss cognitive neuropsychology and specific cases of patients. So this is yet another way in which structure is relevant for function, and this concludes by and large our discussion about neuroanatomy. Now, this is a huge topic, and we can spend the entire course just talking about neuroanatomy. However, this is not the purpose of this course. Rather, this one lecture discussion of neuroanatomy is allowing us to provide a foundation for the study of human cognition and an appreciation of how structure is relevant for function. So with that being said, we're going to move on to the major topic of discussion for today, methods in cognitive neuroscience. And we're going to start by reviewing the list of lecture objectives for today. First, we will look at how neurophysiology helps us gain a fine-grained picture of brain functioning, and also allow us to explore the relationship between animal cognition and human cognition. Then we will proceed to describe the utility of both structural, anatomical neuroimaging techniques and functional neuroimaging techniques that look at brain processing, especially as related, in our case, to cognitive processing and cognitive function. Next, we will examine a number of techniques, how they acquire data, and what are their corresponding advantages and disadvantages. We will look at how the subtraction method has been one of the most popular ones used in functional magnetic resonance imaging, and has played an important historical role in revealing the value of this particular technique in the study of function. Then we will look at how lesion studies complement neuroimaging, and the fact that there is no unique ideal method in itself for studying brain function. Rather, the question that we should be asking in each case is, what is the best method or combination of methods which is optimal for the study of a specific problem in a specific set of circumstances. And last, we will try to conceptualize, or rather illustrate, the value of conceptualizing cognitive processes of interest, let's say working memory, and practice applying this knowledge to a variety of constructs in cognitive neuroscience. Okay, so without further ado, let's get started. And what you may remember from previous lectures is that at the most general level, when we talk about brain function and cognitive abilities, we are talking essentially about the relationship that the brain and the mind allow us to build between the stimulus or the input on the one hand and the response or the output on the other. So we take in information, we process that information. The brain is responsible for this very powerful computational machinery that eventually results in a response, in a motor output. How do we study that? Well, we have to design experiments very carefully. And our first job in that respect is to identify the mental process of interest, to make sure that it is fundamentally involved in the task that we asked our participants to perform. And then we collect a number of behavioral dependent variables such as reaction times and accuracy. This is what typically happens in a behavioral experiment, in a psychological experiment. So here comes in neuroimaging, which allows us to link the brain to various cognitive functions. So what actually happens here is that at the time at which these mental processes are believed to take place, we can also record neural data and try to associate specific mental processes with specific brain processes. For instance, we can ask what part of the brain is responsible for a mental process, when a mental process might unfold, how, and so on and so forth. And we can do this in healthy adults, we can do it in children, we can do it in patients, which overall allow us to get a much more detailed picture of the relationship between anatomy and brain function, including but not limited to cognition. Okay, and the first set of techniques that we should discuss belong to neurophysiology, single or multi-cell neural recordings. And to a large extent, the success and the influence of neurophysiology traces back the relatively simple but very powerful experimental paradigms that try to identify patches of neurons within the visual cortex of non-human primates responsible for processing information from different receptive fields. So what do I mean by that? Well, you can imagine that whenever you look at something, let's call that a fixation point, the entirety of the visual field is projected to different parts of the retina on the back of your eye. So then one early hypothesis in neuroanatomy and neurophysiology was that different receptive fields, different portions of the retina projected different parts of the visual cortex, the primary visual cortex in the occipital lobe, whether in humans or in non-human primates. And the first experiments were conducted in non-human primates. So let's say that a specific cell or group of cells within the primary visual cortex is responsible for processing stimuli within the right top corner of your receptive field. Well, if that happens, then whenever a simple stimulus like a bar of light, let's say, is displayed on a computer screen, then you might expect that cell or group of cells to fire vigorously. And that is exactly what's going on here. The light is presented within the top right corner of the screen, and we can also move it slightly. We can jitter its position. And you can see that the neuron is firing vigorously. We're looking at spikes per second. But then as soon as we move that bar of light to a different position, let's say the right bottom corner of the screen or the left bottom corner of the screen, then that neuron is a lot less active. So what is the significance of this? Well, the significance is, without any exaggeration, huge. This allowed neurophysiologists in the 60s and 70s to map the visual field onto the architecture of the primary visual cortex. What do I mean by that? Well, that means that whenever you look, let's say, at the letter N in neurophysiology on your screen, that information is projected to the center of your visual field, right? A central position on the retina, which projects to this particular areas labeled as 3 and 4 in primary visual cortex of both hemispheres. So 1 and 2 in one hemisphere and 3 and 4 on the other hemisphere. While all the visual information surrounding the center point of your visual field is projected to neighboring areas labeled here as 8, 7, 6, 5, and also as 11, 12, and 9, 10. So a couple of things here that you might notice is the fact that there is an adjacency based on topography. So for instance, 1 here is neighbored by 5, and you see the same organization here. The cortical area responsible for the center of your visual field, numbered as 1 here, is adjacent to 5. The other one is that whatever is processed as the center of your visual field is mapped onto a relatively large patch of cortex in both hemispheres. So the center of your visual field, your fovea, projects to 1, 2, 3, and 4, very large patches of primary visual cortex. And the reason for that is that whatever you're looking at, whatever you pay attention to, is prioritized by the brain, in the sense that it allocates a disproportionately large amount of neural resources to make sure that that information at the center of your visual field receives extensive processing. And we refer to this type of mapping as retinotopic in the field of vision. Okay, so is this type of structured, elegant, and intuitive mapping present only within the visual domain? Not at all. Neurophysiologists have also studied in a lot of detail primary auditory cortex within the temporal lobe. And they found that the principle of organization here is, as you might imagine, no longer space, but frequency. And what I mean by that is that looking at the firing rates corresponding to different frequencies, they uncovered that patches of neurons along the length of the primary auditory cortex respond to increasingly higher frequencies from 500 Hz all the way to 16,000 Hz. And we refer to this as tonotopic mapping. So very elegant, very well structured, and very predictable types of mapping across individuals, but also across species. This is the type of research that initially was conducted in non-human primates and then eventually was confirmed in humans. Okay, so at this stage you might wonder, okay, so this is what happens if we study sensory processing, vision, perception. What about higher level cognitive processes? A classical paradigm in psychology and neuroscience is the delayed match to sample task. And here we have a simple graphical representation of this paradigm. We can see a monkey being invited to view one of two cues, essentially two holes in a small platform, one of which contains a tasty treat, let's say a banana, something that can motivate the monkey, as opposed to a different cue containing something that the monkey might not be motivated by. Okay, so after the cue is visually available to the monkey, a screen blocking visual perception is introduced for a specific delay, for a specific interval of time, which can be one of the variables manipulated. And after that, the monkey is allowed to make a response, to select one of the two cues, which have been conveniently covered by an occluder. So as you might expect, the monkey is quite good at remembering where the tasty treat might be. So if the treat is on the plus side of the platform, then the monkey will usually have no difficulty reaching to grab that tasty treat after removing the occluder. So this is not what's so interesting. Rather, what is interesting is what group of neurons might be responsible for holding that information in memory. Imagine that if you have to preserve the special location of the tasty treat in mind, then a group of neurons should be able to support that type of cognitive functioning. And it turns out that a group of neurons within the frontal lobe of the monkey becomes particularly active during the delay period. They are relatively quiet during the cue and the response phase, but they are especially active during the delay. So based on that, we would infer that this group of frontal neurons are responsible for maintenance of a special location using working memory. Okay, so this is just one illustration of how neurophysiology can track down not only sensory perceptual processes, but also higher level, in this case, memory processes. So this is more or less the technique, and this is its utility. But what about its disadvantages? And one of them is incredibly apparent. If neurophysiology is looking at single cell recordings one by one, or even groups of small cells one at a time, then this is really just drops in a bucket. It's like trying to make sense of a movie we're watching on a screen while only witnessing the values of several pixels on that computer screen. We have 86 billion neurons, more or less, within the human adult brain. So how are we to make sense of the entirety of the brain function if we can only look at several cells at a time? And neurophysiology attempts to deal to some extent with that issue by means of lesion studies. So what that means is that specific parts of the brain of variable sizes and at variable locations can be damaged in animals, and we can witness the impact on behavior. So for instance, something that's been extensively studied is the impact of parietal lobe lesions. And what's been quickly realized is the fact that if an animal sustains such lobe lesions, it is quite typical to develop visual motor impairments. And what that refers to is the ability of vision and motor control to cross-talk in the service of specific actions. So let's say I'm looking right now at my coffee cup and I'm trying to figure out where its position in space it is relative to my body, so I can touch it, grab it, lift it, and bring it up to my mouth. Well, there's a complex chain of information that links vision, telling me exactly where the cup is at any given moment, and motor control, telling me where to reach, how hard to grasp, how much muscle activity I need to elicit to lift the cup smoothly off the table. And this is something that a lot of neuroscientists have spent years of their life trying to understand, and they still do, including here at UTSC nowadays. So this is something that is no longer functioning properly in the context of parietal lobe lesions, which make us believe that the parietal lobe is essential for visual motor coordination. Okay, what other things have we learned about damaging, let's say, other parts of the animal cortex or subcortical structures? So let's look at something very different, and this is the impact of amygdala and temporal lesions on both sides of the brain. And one of the outcomes of this type of lesions is a very bizarre syndrome called Kluver-Bucy syndrome, which gives rise to what we might consider atypical abnormal behaviors in animals. So in this case, you can see a dog trying to copulate with a pig, which is definitely something we do not necessarily consider to be a normal behavior within this species. So different parts of the animal brain appear to be in charge of governing different aspects of cognitive function and behavior. Okay, so with that being said, let's move on to humans and cognitive neuroimaging. The field of cognitive neuroimaging is quite extensive, quite large, and it generally covers a broad set of brain scanning techniques that are aimed at clarifying cognitive function and the underlying structure. Now, to a large extent, this has been driven by clinical users. For instance, identifying the pathophysiology resulting from illness or injury, whether you've been in a car accident or in a sports accident, whether you're suffering from a certain brain infection or from stroke. This is very useful in being able to identify the location of the damage and also the gravity, how pervasive and how badly a part of your brain or a certain brain structure has been impacted. And now this is primarily used for the purpose of anatomical characterization and localization, but occasionally it is also accompanied by descriptions of functional disruption. So, for instance, it might not suffice to know that one part of cerebral cortex, for instance, has been damaged, but we might also be interested to figure out whether remaining function has been preserved in a patch of damaged tissue. And just by looking at an anatomical scan, we're not going to be able to answer that question. So running additional scans that rely, let's say, on functional magnetic resonance imaging might be able to clarify that picture and provide an answer to the question, does, let's say, some cognitive function still is still housed within that damaged patch of cortex? Do we still see that patch of cortex coming into play for its designated purpose, for its typical function? So this is how neuroimaging is used for clinical purposes, but for the purposes of our course, we talk about experimental basic research uses. And this concerns both structural analysis and functional analysis, right? So for instance, we might wish to see how extensive visual and motor expertise change brain anatomy, right? How a particular fiber of axons, how a particular tract is changed, how it further develops in response to the constraints of, let's say, playing a demanding sport, right? That requires a lot of visual acuity and fast movements. Or, and this is a lot more common, looking at functional analysis, how and what brain areas come into play when different mental processes are being elicited within the healthy or altered human brain. And let's start our discussion with computerized axial tomography, also commonly referred to as CT scan or CAT scan. And this is for historical reasons. This was responsible essentially for a revolution in diagnosis, especially in emergency medicine. And the technique nowadays is considered to be quite simple. Just like X-ray is relying on the body's ability to absorb radiation, and depending on the tissue that is being imaged, to absorb it to a different extent. So this is not necessarily a technique limited to brain scans. It can be generally applied to most of the human body. For instance, you can look at the abdomen, you can look at the knee, and again, depending on what tissue we're looking at, the level of absorption will be different, and that will be imaged within the resulting pictures. So as a result of that, for instance, if we're looking at an early CT scan, we can see, for instance, that the skull and the rest of the brain absorb radiation differently. That is quite obvious. We can also see the ventricles in this picture. If we had extensive damage to a part of the cortex, that might also become apparent. And this is very useful, for instance, if we're trying to find out that a part of the brain, for instance, is swelling, right, which necessitates a really rapid and efficient course of action, for instance, a craniotomy, cutting into the skull to be able to remove or alleviate some of the pressure. But this is a very coarse picture. And while CT technology has increased, has become a lot better over the years, still we need better images to assess and to provide a basis for diagnosis. And for that reason, MRI, structural MRI, which we will discuss in a second, is a lot more popular. However, that is not to say that CT scans are no longer used. They are. There are many reasons for which an individual, a patient, could not be placed within an MRI scanner. And when this is the case, then a CT scan can provide a viable and informative alternative. Another problem with a CT scan is the fact that it relies on radiation. And while undergoing a CT scan once or twice is believed not to be damaging, then extensive exposure to radiation through many CT scans could lead to some problems, can raise health concerns. So CT scans have played a historical role in consolidating the value of imaging in the field of structural imaging, but at the same time it suffers from a number of limitations which led to the rise of structural MRI. And this is the next topic we will discuss. So what I mean by structural magnetic resonance imaging is essentially looking at the anatomy of the brain, looking at brain structure as opposed to functioning. And to avoid the confusion, many times when people talk about magnetic resonance imaging, they refer exclusive or primarily to structural MRI. Whenever we talk about function, people prefer to preface that with the letter F. So that would be fMRI as opposed to just MRI or SMRI. Now, if this were a course dedicated exclusively to neuroimaging, probably we would have an entire lecture dedicated just to the physics of MRI. However, we do not have that luxury here. So this is just a Cliff Notes version of how an MRI scanner acquires information. And to build an intuition of what a magnetic resonance imaging machine does, you can think of a giant magnet, a very powerful magnet. And informally, we often refer to an MRI machine simply as a magnet. What does this magnet do? Well, it essentially places the human body, including the brain, within a very powerful magnetic field. We measure the strength of that magnet in Teslas. The most typical magnet nowadays has a 3 Tesla magnitude. And just to gain an intuition about how powerful that is, it's about 60,000 times stronger than the magnetic field of the Earth. Okay, so very powerful magnet. Now, how do we move from placing the body within this magnetic field to acquiring brain or body images? So just like the CT scanner, the MRI can be used to acquire images of the brain, but also of a whole other number of body parts. Well, the trick here resides into a specific mechanism that uses radio frequency to alter the orientation of protons within the human body, within tissue. So let's take a more detailed look at what exactly happens here. When we place the human body within the very powerful magnetic field of the scanner, what happens is that protons, which are randomly distributed in their orientation of spinning, become aligned with the magnetic field of the scanner. Okay, so at this point, a radio frequency coil is typically involved to disrupt the level of alignment of these protons. And also commonly the radio frequency pulse is applied to change the level of orientation to a perpendicular one relative to the orientation generated by the main magnet. But this also has another effect, and that is of building up the level of energy within these protons. So at the same time, it changes their orientation and it leads to an elevated energy state. Okay, so this is the second step. And the third one is simply elicited by turning off the pulse for a brief moment of time. And what happens at that time is that the protons release this buildup of energy as they spin back to the original, to the main orientation provided by the main magnet. Okay, so herein lies the trick, because different tissues of the brain, in our case, relieve different amounts of energy. And that can be used to essentially fill in the brain picture that we wish to take. And it's not just about brain tissue. So for instance, within the skull, we would have a different number of protons releasing energy versus the brain. And also the meninges will have a different amount of energy as well. So depending on what part of the body you are looking at, what part of tissue, different levels of energy are released, and we call that resonance, hence the name resonance within MRI. As a result of which we can take very detailed pictures of the human body. And let's appreciate how detailed that picture is by comparing a CT scan, an early CT scan, with an anatomical, with a structural MRI scan. So as previously mentioned, you can appreciate the difference between the skull and the brain, but you can also see the meninges here, right? You can see the clear distinction between gray matter and white matter. You can appreciate the gyri and the sulci. You can look at ventricles in a lot more detail, and so on and so forth. So high resolution, high spatial resolution images provided by magnetic resonance imaging, which are incredibly valuable to visualize both the healthy, normal brain, but also to visualize potential damage to the human brain in different areas, whether to gray matter or to white matter. And this is orders of magnitude better than what a CT scan could provide for us. Now, it is also exceptional. It is remarkable, the fact that we can acquire high resolution images of the brain within a brief 90-second MRI sequence. So we can just acquire high detail images of the brain within a very brief interval of 90 seconds of scanning. And as we previously mentioned, these 3D volumes of the brain can be navigated then in multiple directions, three of which you might remember are the axial plane, the coronal plane, and this profile view, the sagittal view of the brain. So we can navigate in all three different directions, and usually when we look at a particular cortical area or subcortical structure or white matter, we like to examine this from multiple views to gain a much more complete understanding of how it looks like, whether it is normally developed, or whether it is the locus of an injury, and so on and so forth. So now, since we emphasized so much the level of spatial resolution, the level of spatial detail that the MRI provides, the question is, how can we make this better? And one essential aspect or parameter is provided by the strength of the magnetic field. So most magnets in use nowadays are 3 Tesla magnets, but 70 magnets have also become increasingly popular. And this is because of the kind of spatial resolution increase that they provide. So look at the level of detail that you can get from such a picture. And let's look at another one here. you can appreciate the fine detail, all the level of branching of the cerebellum that is clearly visible from a 7D magnet. Now, there are pluses and minuses with using a 7D versus a 3D magnet, and those pluses and minuses belong to a discussion that focuses on the physics of MRI, which we do not have time for here, but suffice it to say that it is often the case that 7D images are further accompanied by 3D images of the brain, and they can sometimes provide complementary information about brain structure, and as we'll see, have the opportunity to see soon enough for a functional magnetic resonance imaging as well. I also want to mention the fact that UFT has been housing for the last couple of years a new 3D machine downtown, and many of our labs, including several of our labs here at UTSC, currently collect neuroimaging data using this scanner. So this has been a very important acquisition for UFT, and also for GTA at large, since several other universities and hospitals within Toronto and the Toronto greater area are currently using this scan both for research and clinical purposes. So a very important addition to the set of equipment and labs and overall advantages that we have by researching the human nervous system here at UFT. Now, one of the terms that come up a lot within the MRI jargon is voxel. What does that mean? Well, it relates to something that I already mentioned, spatial resolution. That is the level of spatial detail provided by a brain scan. Just like an image is an organized collection of pixels on your computer screen, a volumetric scan of the brain, one in 3 directions, is a collection of organized voxels. And that's exactly what a voxel takes its name from. It is a volumetric pixel. You can also see here that the matrix size of the slice is 64 by 64. So that means that we essentially can view the brain in 64 levels at 50 divided into 64 by 64 such voxels, and they are at this time only viewed in one plane because we cannot visualize properly the 3D structure of the brain on a computer screen. But you can imagine that we can run through multiple such slices of the brain, axial slices in this case, and we can see each one of them representing the brain as a 64 by 64 image. And while this might be quite large, because you can imagine that a voxel of 3 millimeters by 3 millimeters by 3 millimeters is quite substantial. It contains a significant number of neurons. Even that can be quite useful for a number of purposes. So, for instance, a common use of measurements based on voxels is correlating brain region size with neurobehavioral phenomena. So let's say we wish to quantify the relationship between hippocampal volume in number of voxels with PTSD severity of a combat exposed individual. And perhaps many clinicians and neuroscientists will not be surprised to find a negative correlation in this sense. Reduction in the size of the hippocampus as a result of PTSD severity. And here that is expressed in a different unit of volume, but once we know exactly what the voxel size is, then we have the ability to quantify it using slightly different measures. Okay, so this is one particular use. Another one that is perhaps even more common is for the purpose of lesion assessment. And typically when we assess a lesion carefully, we want voxel sizes which are a lot smaller, right? So something like 0.89 isotropic voxels are very common in structural MRI. And when I say isotropic, I mean voxels that have the same dimension in all three directions. So left, right, up, down, and front, back. And what we can do with this is take a very good look at the size of a lesion and try to quantify it in numbers of voxels that do not display correctly. And that happens essentially because that particular patch of gray matter, in this case white matter as well, has been knocked out presumably as a result of an injury or of a disease. And this does not tell us anything about the severity of the injury right away, but it is quite informative to quantify its size, the size of the part of the brain that has been damaged. So this is what happens within a single individual. So then one of the natural questions here is, how do we tend to make use of this in the context of a population of patients? Can we somehow corroborate that information across multiple individuals? Can we run group analysis? And the possibility to do that exists. For instance, the lesion overlap method is quite common in this sense. We can acquire images, MRI images of brain injuries and impairments across a population that has been damaged, and the damage has resulted into some common mental faculty or ability being impaired. So let's say that our patient has started to suffer memory deficits. Then what we can do is overlap the injury of patients with memory deficits. The more patients, the better. And then try to visualize the probability of injury, or the number of patients, that would be the simplest measure, that have a particular area of the brain damage. What specific voxels have been damaged across these individuals. And the color here codes the probability of damage to an area, or the proportion of patients that has that area damaged. So somebody might be quick to point out then that a reliable relationship might exist between the damage to the area shown here in red and a specific type of memory deficit, which is common across these individuals. So I hope the logic of this method is quite is easily apparent, and this is just one instance of how we can make use of information acquired using structural MRI across many individuals to get a better picture of function and its relationship to anatomy, to structure. So with that being said, I should emphasize that MRI has truly revolutionized the field and has been essential to cognitive neuroscientists over the last 2 decades. And for this reason, but not only, it is immensely popular in the news and media. So probably many of you have already had your encounter with MRI in one way or the other. And hopefully this course can provide you with a better view and assessment of what MRI can do for us in terms of understanding brain structure and function. Interestingly though, MRI does have a limitation, and that comes to the inability to speak to the structure of white matter, which appears to be quite diffuse and uniform if you look at an MRI scan. But we do know from physiology, from neuroanatomy, that white matter actually reflects a quite complex interconnection and passage of white matter tracts, bundles of axons connecting different cortical regions with each other, with different subcortical structures with the rest of the brain. So then how can we get some insight and visualize those white matter fibers? And to answer that question, we continue to use an MRI machine. However, we used a rather different procedure, relying on a very different property of the brain and of physics actually, and that bears the name of diffusion tensor imaging, or DTI. So what does that do? Well, it essentially tracks the diffusion of water molecules within the brain and infers the direction of the tracts, their special organization, and their location within the human brain. So let's take a better look at that and try to understand exactly how DTI achieves it. Well, the fundamental principle that it relies upon is that of anisotropy. What that means is that if a molecule is left unrestricted, it will have the opportunity to diffuse with equal likelihood in all directions. However, if its passage is constrained or limited in some respect, then anisotropy happens. And we call it fractional anisotropy because it is typically quantified between 0 and 1, where 0 would be complete unrestriction and 1 would be complete restriction of a water molecule, with intermediate levels reflecting a more likelihood of passage, of diffusion in one direction or set of directions rather than others. So what a diffusion tensor imaging technique allows us to do is to track the degree of fractional anisotropy for every single voxel in the brain, and then try to infer where the white matter fibers are. And this happens because water molecules are a lot more likely to flow or to diffuse along an axon than cross axons. So its passage is restricted in some directions and much less so in other directions. So the most likely direction of diffusion can be used to speak to the direction of a bundle of axons. And by acquiring this degree of fractional anisotropy on a voxel by voxel basis, we can put together a brain map by some complex mathematical techniques that illustrate the complete picture of the brain with respect to what areas are connected with each other. Two other terms bear mentioning here and some explanation. One is tensor, and that is just a mathematical construct, a multidimensional description of what happens at the level of a single voxel. You can think of it as an extension of the idea of vectors and matrices from linear algebra, in case that is familiar to you. But it is somewhat more complex. It can actually be used to link different vector spaces and even more complex mathematical constructs. For us, this is of specific interest here since it is used to model the level of anisotropy, hence the name of the technique, diffusion tensor imaging. The other term that you might come across quite often when talking about white matter tracts is tractography, and I think the name speaks for itself. Is a set of modeling techniques that allow us to move from description of voxels in terms of fractional anisotropy to this presumed descriptions of location and connectivity of different brain areas. In other words, the presumed location and profile of the tracts of the brain, of the white matter tracts of the brain. And for illustration purposes, this is the inferior longitudinal fasciculus, the longest fiber tract in the human brain that runs all the way from the occipital cortex through the temporal lobe to the frontal cortex. So this is huge, and it is also hugely important for visual perception, as we came to know, along with a host of other cognitive functions. Its damage has been recently linked to a whole range of perceptual and memory problems. However, this is just one of many tracts. And here's a bit of a more comprehensive and colorful picture of these tracts. So to single out one class of tracts, you can look at the blue ones here, which serve to delineate the connectivity of the cerebellum with the rest of the brain, right? So you can see how the cerebellum connects to quite a few different cortical areas. And again, you can use different ways of color coding this information to reveal different properties or types of organization across many fibers running through the human brain. Why is this important? Well, it is important for a number of reasons, both clinical and experimental research purposes. So, for instance, over the last 10 years or so, it became increasingly apparent that many types of damage to the brain are reflected not necessarily at the level of gray matter insults, but rather impacting the structural integrity of white matter fibers. So here's one such example from a study. It was found that one fiber that connects the thalamus to the frontal lobe is quite likely to be damaged at a specific location. The position of, or rather the location of the white matter fiber at when crossing into the corpus callosum. And this is very relevant. This is relevant because it can speak to some of the behavioral abnormalities and problems that result from brain damage and cannot be solved simply by evaluating gray matter integrity. You can imagine that the frontal lobe and the thalamus might seem to function quite well, but if the connectivity between them has been damaged in some way or restricted, then they might not be doing everything that they need to do, right? So while structurally they may be intact, functionally they may not be operating properly. And this is very, very important to know. So this is what happened in concussion, and this is by no means the only fiber impacted by this type of damage to the brain. Of course, what specific fiber tract is damaged also depends on the direction of the concussion and the strength and the anatomical variability across individuals and many other factors that need to be taken into account. But again, this just illustrates the power of diffusion tensor imaging as a complement to structural MRI to shed light on both anatomy by way of neuroimaging and as a research tool when investigating different types of cognitive impairment resulting from disease or accidents, injuries. Okay, so far our discussion has been very, to a large extent, focused on structure, right? On neuroanatomy and the ability of imaging to reveal the structure of the human brain, gray matter, white matter, and so on and so forth. But what about function? And before we move any further, I just want to point out that we reached the midpoint of the lecture. So if any of you feel inclined to take a break, then by all means do so. Otherwise, for those who want to continue along, let's have a quick discussion about electroencephalography or EEG. Now, the EEG is a prime example, a classical example of investigating the brain from a functional standpoint. So again, here, the distinction is between functional analysis versus structural analysis. For the type of techniques that we've discussed so far, like structural MRI and CT scans, the focus was on structure, characterizing the topography of the brain for clinical purposes or for research purposes. In contrast, functional analysis tried to characterize mental processes and the neural activity that supports them. It tries to characterize the spatial, temporal, and more generally the mechanisms by which such processes unfold. Now, EEG is one of the oldest imaging techniques available. You might be surprised to know, and first time when I found out about it during my training in neuroscience, I was certainly surprised. The first EEG, human EEG study was conducted in the 1920s by Hans Berger, who also named this technique as EEG. And animal EEG studies were conducted as early as the end of the 19th century. So this is an ancient technique by current standards. And one might feel inclined to ask, does it have anything to offer nowadays? And here I must confess that for a long time I erred on assuming that it really does not. And many people in neuroscience actually have been placed in this position. There's still a lot of misconception and prejudice on EEG based about EEG because of its history. It's a very long history. However, you might also be surprised to know that EEG nowadays is experiencing a resurgence. And that is driven by two things: advances in the quality and sensitivity of the equipment that picks up neural signals, and also in the complex and sophisticated analytical tools which allow us to get a lot more meaning, to make more sense of the recordings that EEG provide. Okay, so without further ado, what does EEG do? And I believe that this picture can provide you with an intuitive understanding of what an EEG experiment might look like. A number of electrodes are placed on the scalp of a human participant, and their number can be quite variable. Nowadays, it's very common for research purposes to use 64 electrodes, but for practical purposes, sometimes a fewer number is used, and for research, let's say for clinical purposes, you can go up to 128, as shown here in the bottom right corner, or even 256, right? So lots and lots of channels. And the key aspect of this method is that each electrode provides a recording channel. Each electrode indexes or records activity, neural activity, from a relatively large group of neurons. Okay, so herein lies both the advantage and the disadvantage of EEG. First of all, it's got solid temporal resolution. These electrodes pick and can describe the neural signal with a millisecond by millisecond resolution. So we can hardly ask for more from any imaging technique. But EEG is very inaccurate or non-specific when it comes to where the neural signal originates. We do know that it captures the broad and coarse activity of a large number of neurons spread out through a relatively large part of the human cortex. But with respect to where that might be, here EEG is not doing so well. Presumably, if the electrode is placed on the forehead or on the front of somebody's head, the origin of that activity is within the frontal lobe, mostly. But when it comes to specifying the precise patch of cortex responsible for it, EEG is faring quite poorly. And this is not necessarily a surprise. Such a trade-off between temporal resolution and spatial resolution is quite characteristic of imaging techniques and methodology nowadays. Okay, so then what does the signal recorded by EEG look like? And every single one of these squiggly lines here can be what one of these electrodes capture, right? So here we've got only several of them, but if you have 64 electrodes, then you're going to end up with 64 such signals. How do you make sense of them? And here is where interesting analytics comes into play. Each of these signals is incredibly noisy. And that happens for a number of reasons. First of all, to capture that activity, the electrode needs to pick up activity that is diffused by the skull. So that severely reduces the quality of the signal, which in the absence of the skull would be much easier to pick up, but we cannot perform a craniotomy on a perfectly healthy subject just to boost the quality of the signal. Secondly, the signal that the EEG is picking up comes from a very broad and relatively diffuse patch of cortex, so even subcortical areas. Then there are limits to the hardware itself, how sensitive it is in picking up a signal. And last but not least, there's a lot of variability from trial to trial in terms of, let's say, how much attention an individual pays attention to, or how distractible somebody is, or how fatigued somebody is over the course of an experiment. So over lots and lots of different trials, these signals can look quite different from each other, even in the context of the same stimulus being presented over and over again. So lots of different sources of noise and variation, both across channels and for the same channel across repeated trials. So then how do we make sense of such a noisy signal? Well, one of the classical techniques with a long and reputed history in the field is based on averaging such signals. So what you can do is present the participant with a particular type of stimulus, let's say a sound. And then in response to that sound, you collect the EEG recording for one channel, and you repeat this across many trials. Let's say dozens, if not hundreds of trials. So again, what happens is that every single trial has, for this specific electrode, elicits a rather noisy signal. So we cannot really make heads or tails of it, but if you average all these dozens or hundreds of trials, suddenly you end up with something that looks much cleaner and perhaps is much easier to interpret. Why might that happen? Well, it's in virtue of averaging out noise. So because on some trials the participant might be paying more attention and on others less attention, because on some trials the sound might seem familiar and in other ones it might not. Everything which is not related to the fundamental processing of that stimulus gets averaged out. So then what remains across this process of averaging are just the fundamental, the common components of the signal. So one might expect, for instance, that the type of deflections that you see here within the averaged EEG signal are indicative of sensory processing. So perhaps this may be the first time when the sound is processed within the auditory cortex, and then later deflections, whether up or down from the average signal, can simply reflect things like memory retrieval. Perhaps some sounds appear to be familiar to the participant. So you can start to index the timing of memory processes. One thing that I should mention is that the averaged EEG signal across as little as 2 trials or as many as hundreds of trials bears the name of event-related potential. And I hope this makes sense because what we are doing here is averaging EEG signals and they're all related to a specific event, in this case a stimulus, and that stimulus happens to be, in virtue of this example, a sound. So everything is timed relative to the onset or offset of that stimulus. So in this sense, we're looking at things which are event related as opposed to a EEG signal during resting state. The other thing that I should mention is that these deflections, whether up or down, are called components. And sometimes they bear names, sometimes they do not. But neuroscientists have spent a lot of time trying to make sense, especially of the meaning of these components. Trying to characterize when they occur relative to the stimulus onset, how big the amplitude is, how variable they are within and across individuals, and so on and so forth. But let's try to illustrate this even better by exploring one example. And I should say that EEG is extremely popular within pretty much every field of cognitive neuroscience. And psycholinguistics is by no means an exception. People who study language and the neural basis of language have been using EEG extensively because they care about the temporal properties of language processing. So here's one classical example from a study conducted in the early 2000s. In which the authors try to figure out exactly when semantic content becomes available. And what they did was present participants with sentences that made sense and sentences that did not make sense. So for instance, it makes sense to say about a shirt that it was ironed, but it makes much less sense to say that a thunderstorm was ironed. So they presented participants with lots of such sentences, some of which made sense and some of which did not. And then they averaged the EEG signal for both categories of sentences separately. And in doing so, they managed to derive 2 different waves. These are 2 different ERPs that I used to try and make sense of when a semantic violation might be detected at the neural level. And it so happens that one of these components, called N400, is where the semantic violation was noticed, was recorded. Now we call this N400 because you see this is a negative deflection, and this is just respecting some of the old tradition and conventions in the field where negative values are up and positive values are down. So this is a negative deflection relative to a zero baseline, and also its peak, at least for properly formed meaningful sentences, occurs around 400 milliseconds. Now, when you look at the shape of this ERP for semantic violations, for the second type of sentences, you see that this peak occurs slightly later, but the amplitude of it is significantly larger. Now, we continue to call this the N400 component, right? So that we can assimilate it or relate it to the normal N400 triggered by meaningful sentences. But the thing that we should be paying attention to here is the fact that this large deviation is systematic and well timed relative to the onset or offset of the stimulus, of the sentence that was being heard or read. So what does it mean? Well, this is very important because it tells us that around 400 to 500 milliseconds after you listen to a sentence, you already have access to its meaning, and you can detect whether the meaning is in line with your expectations or it violates your expectations. And if you are a psycholinguist, then you pay a lot of attention to how meaning is extracted from words and sentences, how rapidly, and what neural mechanisms makes that possible. So this is just one example of how ERPs can be used to infer mental chronometry, the timing of mental processes that underlie language. Now, as previously mentioned, EEG, or to use a fuller description, scalp EEG, because the electrodes are placed on the scalp, has one major limitation: its poor spatial resolution. Each channel, each electrode collects information which is the result of summation of electric potentials of thousands, if not millions, of neurons. And this provides us with a very poor understanding of where the signal is coming from. And furthermore, the skull acts as a further filter, disrupting and diffusing that information. So what if we could place the electrodes directly on the skull? What if we could record activity directly from gray matter? And this is indeed done, but never just for research purposes. This is something that needs to be done for certain patients who are ready to undergo surgical procedures, especially in the treatment of epilepsy. And this should bring us back to our conversation about Wilfred Penfield, right? So you remember that in order to trace the origin of epileptic seizures, he had to record activity from the brain at various locations. And nowadays that technique has got a lot better, and the equipment is a lot more sensitive. So here's one picture that might show you what an array of electrodes placed on the surface of the brain looks like. This provides us with a unique opportunity from a research standpoint, because the patient already has this array of electrodes collecting information about neural activity at a specific location. So in this case, we have both the temporal resolution provided by scalp EEG, but in contrast to scalp EEG, we also have the spatial resolution provided by inserting the electrode within a uniquely localized patch of cortex. And typically the spatial resolution is several millimeters for intracranial electrodes. Now, we call this technique electrocorticography, or ECOG, and essentially comes down to placing EEG channels directly on the brain. So we also call this intracranial EEG. And on the right hand side here, you can see an example of what the function underlying or rather supported by neural activity at different locations might be as a function of the type of tasks or stimuli that the participant has to deal with, the patient has to deal with. So for instance, you might know that some electrodes pick up a lot of activity when the participant is engaging in motor control, moving a hand, let's say, or a foot. A lot of them are responsible for sensory signals, and some of them are responsible for speech. So a lot of neural activity detected when the participant is producing speech signals. So very exciting, very powerful, very informative as an imaging technique, but also highly limited. It is invasive. It can produce additional damage, though this is minimized by the development of novel and newer types of hardware, specifically better electrodes inserted into the cortex. But again, no healthy individual probably would be amenable to undergo such a procedure if this was not absolutely necessary for health-related concerns, right? For treating an epileptic seizure, right? Part of a surgery procedure. So it is quite limited in terms of the number of participants who undergo this procedure, and it is also limited in terms of where the electrodes might be placed. So this electrode array is placed at areas that are suspect of eliciting the seizure, or areas that might be necessary to perform surgery across in order to get to the origin of the seizure. So again, lots of limitations, both in terms of where we place the electrodes, what areas we can study, and also in terms of the participants or the patients that can provide this type of data. Now you might wonder, is there a way to gain information about brain function with a decent spatial resolution, but in a less intrusive, in a less invasive manner than ECOG? And historically, that's the role that positron emission tomography, or PET, played. So what does PET do? Well, in a typical PET study, patients or participants are injected with water labeled with a radioactive oxygen isotope. Now, when injected with this isotope into the bloodstream, the result is that the blood supply, the blood flow carries soon enough these isotopes into the brain, where due to their highly unstable nature, these isotopes release positrons. Now, these positrons collide with electrons, which lead to the generation of gamma rays in opposite directions. And this is exactly what PET is sensitive to. PET is essentially a gamma ray radiation detector. It identifies where the source is of this radiation, where the gamma rays have been released from. How is this helping us with brain function? Well, what's important to note here is the fact that areas of the brain that are particularly active at the moment of time have a higher metabolic cost. They require more oxygen, and that oxygen involves more blood flow. Hence, those also happen to be the areas where radiation tends to pick up. Okay, so once this radiation is detected and associated with different parts of the brain, all this information is put together by a computer and can be visualized as images of brain function. PET has been quite popular in the study of cognition throughout the 80s and the 90s, and nowadays continues to be quite popular for clinical purposes. So, for instance, one study that bears particular notice is the result of research conducted at University of Pittsburgh in the study of Alzheimer's disease diagnosis. Now, one of the facts that we do know about Alzheimer's is the fact that a particular type of protein, which naturally occurs in healthy individuals, tends to accumulate in much larger amounts and form plaques within individuals who suffer from Alzheimer's. These beta amyloid plaques achieve such concentrations and accumulations that disrupt the normal functioning of the neurons around them. And this is something that was well documented and known, especially through post-mortem analysis of brain tissue. However, PET allowed the imaging of this beta amyloid plaques for the first time by use of a very clever technique. Specifically, what researchers did was come up with a special radioactive chemical that has affinity for these beta amyloid plaques. And hence imaging the higher levels of interaction with this beta amyloid plaques allowed diagnosis of Alzheimer's and also allowed imaging exactly the location of higher concentration of beta amyloid plaques within the brain. Now, this happened, this specific study was published in 2008, but PET continues to be widely used in the study of Alzheimer's and also in the diagnosis of a variety of types of cancer. However, again, at least two aspects of this technique are readily pointed out as weaknesses. One is the fact that while its utility is beyond controversy, still it requires the submitting the participant to certain levels of radiation. And while this may be fine for 1 or 2 or 3 sessions, repeated exposure to radiation might turn out to have negative impacts on the health of participants in studies, let's say, for research purposes. The other one is that the spatial resolution that you see here, it's still rather limited, so probably you can do better than that. And here enters functional magnetic resonance imaging, or fMRI. Now, fMRI and PET do bear some similarity. And that's a similarity with regard to the general mechanism that they exploit for the purpose of generating brain images. What is the general mechanism? Well, it is the importance of blood, to be more precise, of oxygenated blood for neural activity. We already mentioned the fact that the metabolic cost of brain activity is quite high. Neural activity is supported by oxygen and glucose, and an increase or a correspondingly high level of blood flow to a particular cortical region is responsible for supporting that metabolic cost. Now, PET exploited that with the use of unstable isotopes injected into the bloodstream of a patient or of a participant in a study. However, that was quite invasive, and the question was whether we can exploit that level of oxygenation using a somewhat different technique or property of oxygenated blood. And it so happens that in the 90s, one scientist, Seiji Ogawa, along with others at the time, noticed that the magnetic properties of oxygenated versus deoxygenated hemoglobin are quite different. Now, hemoglobin is a protein that carries oxygen within our blood. So he came to the realization that he can use the difference in magnetic moment between oxygenated versus deoxygenated hemoglobin to track down blood flow and indirectly neural processing and its level. And that is exactly what we are measuring here. When using fMRI, we are tracking down the level of blood oxygenation. And the acronym for that, which you will come across quite often if you delve into the fMRI literature, is BOLD, the BOLD signal, the blood oxygenation level dependent signal. So this is precisely what fMRI keeps track of. This is what it monitors, this is what it registers, and this is essentially what we are looking at when examining an fMRI brain picture. Two things should be mentioned here. One is that fMRI allows us a solid spatial resolution. The MRI machine, which we already know can be used for a variety of purposes, such as DTI and structural MRI, and in this case fMRI, is specifically designed to capture minute local differences in magnetic properties at the level of a tissue, let's say a brain tissue. However, it's got much weaker temporal resolution, right? We are tracking down blood flow. We are not tracking down neural activity in a synchronous manner. And both because of the fact that we're tracking down blood flow and also because of the intrinsic limitations of the MRI techniques, the temporal resolution is at the scale of seconds rather than milliseconds, which is what we saw for EEG, for instance. It is quite common to have a temporal resolution of 2 seconds for many experimental study nowadays and over the last few years. Many of the studies in my lab use a temporal resolution of 2 seconds. It has also become increasingly more common to drop that temporal resolution, to increase it to a second, and sometimes even the sub-second resolution, let's say 3/4 of a second. However, there are some additional costs, there are trade-offs, so we do pay for a higher temporal resolution by sacrificing some other properties of the signal, and that's a discussion for some other time. But also we should know that even a resolution of 3/4 of a second is nowhere close to the millisecond resolution that we can get with EEG or ECOG. The other point that I wish to make is the fact that, again, we are not measuring directly neural activity. It is not as if we inserted an electrode into a patch of cortex and we managed to record activity from a group of neurons or even a single neuron. Rather, what we track down is the blood flow correlate of neural activity. And this is based on a very solid association. The name for that association between blood flow, the increased demand for blood supply, and neural activity, it is referred to as neurovascular coupling. However, as you might know from statistics, no correlation or very few correlations that we find in nature are actually perfect. So we examine the level of neural activity through an imperfect correlate. And that is something that we should be aware of, and it is something that will pop up in our future discussions soon enough. However, despite these limitations, fMRI has quickly become, over the last 20-25 years, the main tool, the main methodology for cognitive neuroscience. And there are a number of reasons why that happened. One is that it is so versatile. We already saw that we can use it for the purpose of structural MRI, DTI, fMRI, and a few other things. It can be used for imaging brain structures, and also it can be used for measuring activity in some, or tracking down the anatomy of other body parts. It is widely available. You can find MRI machines in hospitals, in research institutes, in universities such as our own. So again, a diversity of purposes and versatility in terms of use. Now, the publicity raised around fMRI also helped quite a lot. So probably all of you are already familiar with images such as this from movies, from social media, from pretty much everywhere. It's difficult not to come across such images when talking about neuroscience, whether in research or clinical contexts. And an image such as this is meant typically to emphasize two things. One is brain areas that are responsive to a stimulus or a task, and also differences in levels of activation. So a common color code is to display or to paint with warm colors, red, orange, yellow here, the gray matter and the subcortical structures that are particularly active relative to a reference whenever the subject engages in a given task or is exposed to a set of stimuli. And in contrast to that, we paint with cool colors such as blue and green here, regions that exhibit deactivation, so less activity than you might see while the participant is, let's say, at rest, not being committed to a task or being shown any kind of stimuli. However, this is a very coarse and possibly misleading explanation of what's going on when we're looking at this brain images, and also it might not necessarily help us very much understand what's going on. So let's take another example, slightly more complex, and concretize that situation. Let's say that we're asking someone to think about their friend's face. And this is the kind of image that we get from an fMRI scan. Well, what happens here? Several things. When you're thinking about your friend's face, perception is engaged, right? There's visual perception associated with trying to conjure up into your mind the face of your friend. Memory comes into play, right? You have to remember how your friend looks like. Attention comes into play. When you're trying to imagine something at rest, it's not easy, right? You have to really focus to be able to conjure up in your mind the image of that face. Semantics and language come into place. And this is to a large extent an automatic process. You're thinking about your friend's face, but the name of your friend also comes to mind. And a whole bunch of things about your friend come to mind, such as gender, age, other friends, and so on and so forth. And last but not least, the emotional system comes into play. Perhaps your recent set of experiences with your friend made you feel in some way or another. So lots of different systems associated with cognition and emotion come into play when we think about our friend's face. And the question is, okay, this is a massive system and we see activation and deactivation to a lot of regions, but which ones are actually responsible for a friend's face and which ones are responsible for just general mechanisms involved in most memory processes or most perceptual processes? And that's a hard question to ask. Now, this is a particularly difficult example, and we'll have the opportunity to see that when we talk about faces, this generated quite a lot of debate. How do we study that? How do I identify potentially local sources, specific sources of processing face information or familiar face information? So let's try to answer a rather simpler question. Let's say that we are designing a study involved in or dedicated to color processing, and we're asking a simple but normal question. What are the brain areas that are involved in color processing? So how do we go about doing that? Well, this is where the subtraction method comes into place. Essentially, the idea between this method is that we will be presenting the participants in our study with a set of color images. So color is involved in the types of processing that we might expect when recognizing or just viewing a number of color images. But a lot of other things come into play too, right? So attention again comes into play, object recognition comes into play, because we're not just viewing those images, we are processing their content, their meaning, right? We recognize buildings or other people or animals or landscapes from those images. So now we're trying to figure out what is unique to color processing from this entire map of activations and deactivations that fMRI provides for us. And the key, one of the key aspects of a properly or well-designed fMRI experiment is to come up with a good control state. So let's say we present the participant with some other images, because those other images also recruit attention and object recognition and very basic visual processing. And then by subtracting maps that correspond to color images versus images that are not colored, we might come upon the areas which are specifically responsible for processing color information. So again, just to concretize a bit more our example, let's say that we present a participant with lots and lots of color images. Why lots of images? Well, for two reasons. First, we need a diversity of images, so the brain areas that are recruited by these images are not just in virtue of the specific meaning or association you might make with one or two images, right? But we're trying to establish a much more general mechanism that is responsible usually for processing such images. And also because we need lots and lots of trials to estimate a reliable signal. And that's a point that we made about EEG. Well, it's not a surprise that the same idea also applies to fMRI and to many other neuroimaging modalities. It's very difficult to establish anything just from a single trial. Hence, we need to present lots of images, record activity in response to those images, and then average their result, the brain maps generated for each one of those images. So lots of different images and lots of different trials. We can estimate what happens when the participant looks at these color images. And this is just a cartoon. Things are typically a lot more complicated. But let's say that this area comes into play, and this area seems to come into play as well when we look at these images. Okay, then what's a good control here? Well, a common control for such experiments dedicated to color is grayscale images. So essentially what we're doing is try to design a control which is as close to the original one, but not involving color. So grayscaling the images provides us with a reasonable control in that sense. So now again, we can present the participant with all the images that we've seen that the participant saw before, but in the grayscale version. We can average the activity maps just as before, and let's say that the result of that averaging procedure is activity within this cortical region. Then if we subtract this second map from the first map, we end up with one particular region right here, which corresponds more or less with V4, an area we already know from physiology that is responsible for color processing. And that's one of the regions that we will be discussing in future weeks. Now, again, this is a cartoon. Usually brain maps are a bit noisier than that. But they do convey the main point, the importance of a good control condition. And I would go as far as to say that there is never an ideal, a perfect control, but we can strive to make a control as good and transparent as possible, as easily to interpret. And we should always adopt a critical mindset when interpreting the results of such methodology, so results based on the subtraction method, because a confound can always sneak in. So for instance, let's say that instead of grayscale images of this color stimuli, the participants saw grayscale images of some other objects. Then we would have a confound, because the difference that we see in activation between these two conditions would be introduced not just by color, but by also different sets of meanings. So let's say all the color images were of natural scenes, and all the grayscale images were of blocks, of artifacts, of human artifacts, landmarks, buildings, and so on and so forth. Then there would be a difference at the level of meanings, of semantics, and not just at the level of color processing. And even in this situation, where I carefully controlled for that, other confounds might come into play. And I would even go as far as to challenge you to think about one possible confound, if not more, that could sneak in when I'm designing controls based on grayscale. So again, there's always the possibility of a confound, no matter how carefully and you've designed your stimuli and conditions. So always try to adopt that critical mindset and be critical of your own results and their interpretation. Okay, at this stage we already discussed a number of neuroimaging methods, their pluses, their minuses, but what might be obvious at this point, perhaps to some of you, is the fact that intrinsically there is a limitation to these neuroimaging techniques, and that is their correlational nature. And by that I don't mean just the neurovascular association noted in the case of fMRI, but the much more general type of correlation. It's the correlation between the specific state that a participant is placed in as a result of being exposed to some stimuli, involved in a task, exhibiting signs of a specific disease, and so on and so forth. And on the other hand, the brain images that we acquire using any one of these methodologies, whether EEG, whether fMRI, or even neurophysiological measures. And what is the actual problem with that? Well, the problem is the following. We do know that, let's say, a specific patch of cortex is highly active when the participant is placed or engaged with this task. And perhaps through some careful use of a subtraction technique, we might say that this is targeted at a specific property of the stimuli or of the task. So far, so good. But does that also mean that that particular patch of cortex is critically and causally involved in the processing of those stimuli or in facilitating a given task? Well, I hope all of you remember the old adage, correlation does not equal causation. It could simply be that cortical area is recruited for that task indirectly as a result of the architecture of the brain. But that doesn't mean that if knocked out, the brain will not be able to equally aptly and efficiently process those stimuli or engage with that task. So how might we be able to answer that question? And this takes us back to the importance of neuropsychology, the study of patients with brain injury. And this type, whether the brain injury is acquired through disease or accident, it doesn't really matter as much. What matters is the fact that this time we do have the opportunity to examine the cause and effect relationship. An entire part of the brain has been removed, knocked out, damaged, and then we can examine the cognitive effects on behavioral performance, let's say, on those participants. So again, this time we're moving beyond correlation and we're stepping into the area of causation, which is crucial for drawing powerful inferences, rigorous inferences about the relationship between structure and function. Does that mean that the study of patients with brain injury in neuropsychology is not fraught with controversy and disputes? Not at all. There's still a great deal of difficulty trying to come up with uniquely accepted interpretations of some of the data, and there are many reasons for which. One of them being the fact that brain injuries are so diffuse. It's not as if they take out a very well delimited cortical area, let's say, but instead we are very much the victims of this specific anatomy and of the type of accident or diseases that we suffer from. So injuries typically result in quite diffuse spatial profiles of the damage inflicted upon the brain, and that makes it very difficult to identify precisely what cortical area or subcortical structure, let's say, is responsible for the loss of a faculty or its degradation, whether sudden or progressive. And this takes us to one other technique that we will be discussing today, transcranial magnetic stimulation, or TMS. TMS is one of several techniques that induce a temporary virtual lesion in a human brain in a safe manner. So what is aiming to create is a temporary dysfunction for a relatively well delimited part of the brain. And several aspects make this type of techniques quite valuable. First of all, they're repeatable. It's not as if we can only disable that region once and draw conclusions just based on a single observation, but we can repeatedly induce that virtual lesion and examine changes in behavior and cognitive performance associated with that lesion. Hence, TMS is often referred to as repetitive TMS or RTMS. Another key aspect of this type of technique is that everything happens in a lab setting, which means that we have the luxury of testing the levels of performance which are relevant for what we attempt to do here in a pre-injured state. Okay, so let me clarify that for a moment. Whenever a patient has undergone some kind of brain injury, let's say, the decrement that we see in a certain ability, let's say face recognition, could partly be attributed to the injury, but perhaps the participant was quite bad at face recognition to begin with. Hence, it's not quite clear the extent to which a decrement in performance or the loss of an ability is actually due to injury, and to what extent it is due to prior issues, problems, or difficulties that an individual suffered from. Okay, so in contrast to that, in an experiment, we can constantly go back and forth. We can evaluate the performance with respect to face processing before the virtual lesion is induced and after. And we can repeat this across many trials to acquire a sufficient number of observations across which, by processes like averaging, we can obtain more reliable estimates of what, how, and also how much has been damaged or altered as a result of disabling the normal processing within a part of the brain. Now, what TMS does to this end is apply a temporary magnetic field. So we have a magnetic coil through which a changing electric current induces magnetic fields which disable cortical activity within our test subjects. Now in the picture here, the magnetic field and the disruption is primarily induced at the level of the occipital cortex at the back of the head, right? So you might expect that our participant would experience difficulties with processing visual information within the occipital lobe. And this is indeed the case. So here's one way in which we could illustrate the benefits of this type of approach. Let's say that we temporarily disable processing within the occipital lobe by applying TMS signal. And what we vary here is the precise moment when a TMS signal is generated. So let's say the participant views a stimulus, and then I can deliver this disruptive signal right at the moment when the stimulus was presented at 0, or possibly before or after 20 milliseconds, 40, 60, 100 milliseconds, 200 milliseconds. And we can trace the impact that the precise moment when the signal was delivered has on behavioral performance. Let's say the ability to report correctly what the participant saw on the screen, in this case, the letter K. And if you construct a plot of behavioral performance, in this case accuracy, over time, what becomes apparent is the fact that the disruptive signal has an impact on behavioral performance when the signal is delivered roughly between 70 to 130 milliseconds after stimulus onset. What does it mean? Well, at face value, it means that the critical involvement of the occipital lobe in visual processing associated with a stimulus, such as a letter, occurs around 70 to 130 milliseconds after the stimulus is visually presented. So why not immediately, you might wonder? Well, for a number of reasons. So you might realize that it takes a bit of time for the neural signal to reach the back of your brain, the occipital lobe, after going from the retina, passing through subcortical structures, all the way to the gray matter at the occipital lobe. So being able to quantify that precisely with the use of TMS is valuable. And we might be able to draw a very similar conclusion using EEG, for instance, or even ECOG. But again, there what we witness is primarily a correlational effect, right? So if we we have the advantage of creating a virtual lesion with TMS, we can actually establish a causal role of the occipital lobe in processing visual information. Now, one graphical display that you often encounter in neuroscience textbooks whenever discussing neuroimaging techniques is the following, or rather, this is one version of such a display. And the goal of this display is to make clear the trade-off between the spatial and temporal resolution of some of the most common, most prominent neuroimaging methods used in neuroscience. For instance, we already discussed that ERPs exhibit very good temporal resolution, right, at the level of milliseconds, but rather poor spatial resolution. So with ERPs, we're not doing quite so well in terms of identifying the source of the signal that generated the signal of interest. In contrast, functional fMRI, shown here, appears to trade off the temporal resolution in favor of spatial resolution. There are many other methods here that we haven't had the opportunity to discuss yet, and we cannot possibly cover the entirety of methods used in cognitive neuroscience. There's also a constant influx of new methods that are being developed and evaluated, so the field is constantly changing in this sense, and also trying to adjust or modify the borders and the sensitivity of the techniques displayed here, both in space and time. An important message of this type of display is the fact that no method is best overall. Instead, whenever we study a cognitive process of interest, we have to identify the method that is the most adequate for its particular study. And it is often the case that by engaging the use of many different techniques, we can gain a much more fine-grained and informed picture of the neural basis of a cognitive process. One other thing that I should mention here is that this is just one way of evaluating methods in neuroscience. So for instance, if you're looking at this and you see naturally occurring lesions with poor temporal and spatial resolution, you might conclude that they're not so useful overall. However, again, this is just one way of portraying the pluses and minuses of these methods. We already shown or discussed that naturally occurring lesions as well as induced lesions have clear benefits in terms of clarifying the causal role of certain brain structures in facilitating cognition.